Hello, everyone. Welcome to the fourth seminar in the Deakin Science and Society Network Healthy Futures series. My name is Professor Jeff Craig, and I lead the Early Life Epigenetics group within the Impact Institute at the School of Medicine at Deakin University. I also lead the Gene Equality Deakin University Research Network. My Gene Equality colleague, Associate Professor Neera Bhatia, and I are the organizers of this Healthy Futures seminar series. A bit about the Science and Society Network. It was formed in 2018 in recognition that scientists and humanities and social science researchers need to work together to meet the great challenges of the century. No single academic field can bring about the changes that we need to see in the world. Bridging disciplinary divides is the key to finding new solutions to the problems we all face. The Science and Society Network supports early and mid-career researchers across the university who are embarking on groundbreaking interdisciplinary projects. I would like to begin by acknowledging and paying my respects to the traditional custodians of the land on which each of us are today. I would like to pay my respects to the elders, past, present, and emerging. Let us pause to learn, acknowledge, and celebrate their ancient histories and cultures. For those that have not attended one of our seminars today, quick rundown on the structure of the event today. I'll introduce our esteemed speaker, Professor Oded Rechavi. Oded will present for about 40 minutes, and then we'll have some discussion comments from Anthony Hannon and Elsha Lawson-Boyd. And then we'll throw to general discussion questions from the audience. So I'll be chairing the discussion on Zoom. Tarfan will be moderating the YouTube live chat where you all are at the moment. I'm keen to hear answers to your questions, so you can either post questions into the YouTube chat, send us on Twitter at SSN Deakin. Make sure that you use the hashtag SSN Seminar and hashtag Healthy Futures, or email ssn-info@deakin.edu.au. So I'm absolutely delighted to introduce Professor Oded Rechavi as a full professor in the Life Sciences faculty at Tel Aviv University. He has a mission, and it is to challenge fundamental long-held scientific dogmas. Using C.elegans nematodes, he provided direct evidence that an acquired trait can be inherited, worked to elucidate the mechanism and rules of small RNA-mediated transgenerational inheritance, discovered that nematodes' brains can control the behavior of their progeny, and identified a simple neuronal circuit-level mechanism that explains economic irrationality. Aside from his work on nematodes, Oded utilized genome sequencing of ancient DNA to piece together fragments of the Dead Sea Scrolls and demonstrated toxoplasma parasites can be genetically engineered to deliver drugs to the nervous system. Oded has won many prestigious awards and was selected as one of the 40 most promising people in Israel under 40. On a personal level, I came across Oded on Twitter, through which he recruited speakers to a physiologically irrelevant conference in Tel Aviv in 2020, and also known as the Woodstock of biology. The conference was informal, and speakers could choose walk-up theme songs, something that I'd love to do one day. I'm still thinking what mine would be. He's also become the master of research memes on Twitter. So please check him out at Oded Rechavi. So with no further ado, I think Baruch Haba. Welcome, Oded, and over to you.
Thanks a lot, Jeff, and thanks for the introduction and for inviting me to give this talk. It's a real pleasure. Can you see my screen now? Yeah. Great, excellent. Yeah, so I wish I could have been. I could visit Australia again. I hope to do it in the future. I was there once 4 years ago, 5 years ago. It was amazing, and I really need to go back. But this is also great, and I'm very happy to be here. And to present in this forum, which I understand includes also people from the humanities and from very different fields. So I will tell you a little bit about what we do and from a broader perspective, but of course, later you are very welcome to ask me anything in any detail. Okay, so the topic of the talk today is memory and memory inheritance. And we will start from the psychological or anecdotal more, and then we'll go to the details and to the molecular. Already in the 80s, there were studies that showed that we are not born a blank slate. And this is, of course, something that we know from philosophy. It was discussed by the Greeks for a long time. But these experiments that were done in the 80s in the United States were conducted with monkeys. And what they did there is they examined monkeys that were born in captivity in the lab. So they don't know anything about the outside world. And they found that these monkeys have a very hard time learning that a toy flower is scary. So if you pair a toy flower to something scary like an electric shock or a loud noise or something like that, eventually the monkey will understand that the toy flower is scary, but it will take a while. You'll need to pair the toy flower with the scary element multiple times until they learn. In contrast, while monkeys are not born afraid of snakes, it's very easy to teach them that a toy snake is scary. So you just need to pair the toy snake with the shock once, and they already know for life that it is scary. So they have it's much easier for them to learn that snakes are scary than flowers. And the question is why, and how is this memory encoded in the heritable materials? Because they are born with it. They are born with this tendency to be afraid of one thing and not the other. And this, I think, is a very, very interesting question. The problem is that it's extremely difficult to study. And one of the reasons that it's difficult to study is that our brains, and also monkeys' brains, and also mice's brains, are very complex. So our brain has billions of neurons, brain cells that are connected. Each of them is connected to other neurons by thousands of synapses. So the numbers are just totally astronomical. And to make things worse, it also changes all the time. So it's very dynamic, the connections are. And even if I had an identical twin, his brain would be wired completely differently than mine. This is why when I approached this problem, I chose to study a much simpler organism, a much simpler animal, which is called C.elegans. And these are simple nematodes, tiny microscopic worms that you can't see by eye, which are very important model organisms for science, for genetics, and also for neuroscience. And one of the advantages of using these worms is that they have just 302 neurons. So their brain or nervous system is extremely simple in comparison to ours. Altogether, they have less than 7,000 synapses. And moreover, it's always the same. So the identical worms are wired in the same way. And we know each of their neurons by name. So we have access to each of these 302 neurons, and we know them, and we know which neurons they talk to. Despite the simplicity of the worm, it can still do many complex behavior. So obviously it doesn't have the cognition and emotions that we have, but it can still sense, for example, thousands of odors, and mate, and sense temperature, and even learn very simple things, for example, associations between food and odors. They can hold these memories for a few hours before they forget. And some memories can even stay longer. And we have a map of the nervous system of the worm, which is like a subway map, a connectome. We have this since the 80s. So we know exactly with which neurons each neurons talk, and how many synapses are formed between each neuron pair. And this is amazing. We can also activate the neurons or silence them at will using light and other techniques. And we can activate the worms and using light and just make them drive like a remote control car. So this is really an unbelievable level of understanding and control. And the trade-off is, of course, their simplicity. So I thought that this would be a good model system to study memory in. And this is what I wanted to do when I went to do my postdoc at Oliver Roberts' lab in Columbia University at 2010. I wanted to study a particular type of memory, which is even more defined and specific, and this is called imprinting. Imprinting is a long-term memory that can only be acquired during a specific stage of your life, normally early on in life. So you probably heard about the salmons learning to recognize the place where they were born and swimming against the currents to reach the same place, or the neonatal attachment, the strong attachment that is formed between a baby and its mother. These are forms of imprinting. Imprinting was studied in the originally discovered and described by Konrad Lorenz, who got the Nobel Prize for that. And he studied birds and showed that birds will be imprinted. They will learn to follow the first moving stimuli that they encounter in life. So normally it's the mother, but it can also be, you see in this case, a dog or a kid or anything else that will be moving. And this attachment that is formed between the newborn and this moving stimuli is very strong and long lasting. Now, also in C.elegans, Oliver Hobert's lab, in collaboration with JeanJacques Remy's lab, have shown in 2005 in a famous science paper that worms can be imprinted. So normally worms forget. If you teach them an association between food and odor, they will forget after 2 hours. Food for worms are bacteria, E. coli bacteria that we can cultivate them with. However, if you make this pairing, if you take a neutral odor that doesn't mean anything to the worm, and you pair it to bacteria, but you do this, that is good, that is food, you do this pairing early on in a specific stage of development. It's called the L1 stage. So worms move through 4 larval stages. If you do it in the first larval stage, you do the pairing, then the worms learn to associate the odor, this otherwise naive odor, with the food, and they remember that it's appetitive, that it's attractive for them for the rest of their lives, which is a few weeks. The power of C.elegans, and this is why I wanted to study this in Oliver's lab, is that you can actually understand the mechanism. And indeed, they found that this is dependent on one particular neuron and one receptor. So the resolution of understanding is very high. However, when I reached Oliver's lab to do this was in 2010, just when I got to his lab, JeanJacques Remy, you see the first author of this paper, who works in France, published another paper which was even more amazing, that completely sidetracked my development as a scientist. And what he published in his current biology paper is the observation that not only do worms get imprinted, but that the descendants who never experienced the pairing of the odor with the food also remember, and also their descendants, so on. So the memory transfers transgenerationally for multiple generations. And this was a non-mechanistic paper. This is the only figure in the paper, only this skin that you are seeing here. So it was very mysterious and wasn't clear how this is happening. This is just a sign of amazement because the monkey is, because this is not only unintuitive, the possibility that we can learn something and transmit the memory to the next generation, it also stands against one of the most basic dogmas of biology. This is why this paper is still very controversial till this day. The dogma that it violates is called the Weizmann barrier. So in the 19th century, August Weizmann, which was a very famous and important, extremely important biologist, formulated this law. He didn't use these words that you see here on the page because these words didn't exist. But what August Weizmann said was that germ cells are the only cells that can transfer information, genetic information, to the next generation. And then the germline, the sperm and the egg, the germline is isolated from the rest of the body, from the somatic cells. So what happens in the soma stays in the soma. This is called the Weizmann barrier, this separation. And Weizmann formulated this law, which is now also known as the second law of biology, to show that Lamarckian evolution, the evolution of Jean-Baptiste Lamarck, cannot be possible. And I will not elaborate too much about Lamarckian evolution. I will just say that this is considered to be an explanation for evolution which is totally wrong, in contrast to Darwin's natural selection. Lamarck had an earlier theory of evolution, and what he suggested is that acquired traits are transmitted across generations. You probably heard the example about the giraffe's long necks, that according to Lamarck, the giraffe has a long neck because its ancestors extended their necks toward the trees and transmitted this change to the next generation, which is totally different from Darwin's explanation of natural selection. We know that Lamarckian inheritance in its naive form is wrong because because we can't just, what happens in our body doesn't transfer to the next generation. If you go to the gym and you build up muscles, your kids won't be stronger as a result. This is trivial, okay? Now, the reason that only the germ cells can transmit information to the next generation is that only the germ cells can transfer their DNA to build up the next generation. So if you have a mutation, every cell in our body has the same DNA sequence, the entire genome. But if you have a mutation in the genome, in your muscle cells, for example, there's no way to communicate this genetic change to the germ cells. The mutation will stay in the muscles. It won't affect the germline genome, and therefore the next generation would develop as usual. Okay? So the Weizmann barrier is compatible with modern genetics, with modern DNA-based genetics. So when I started working on this and thinking about the imprinting and also in general, more general about the possibility of transmission of memories between generations, I thought, I went back to the molecules and I thought which type of information carrying molecule could potentially transmit parental responses or memories from the soma to the germline and across generation, breaching Weizmann's barrier. And I knew the DNA can't do it. Changes in the DNA can't move between the soma and the germline. And the molecule that I thought about was RNA. And now, of course, because of the Pfizer and Moderna vaccines, everyone knows about RNA, about messenger RNA, which is this intermediate between DNA and proteins. But just for the sake of people from the humanities who don't know, I'll explain it in one slide, just how it works very, very briefly. You can think about the DNA as the IKEA book with instruction to build all the furniture and everything that you need in your house. And you can think of the messenger RNA as the instructions for one particular furniture or for a plant or whatever that you need. In every different cell in your body, just like you need different proteins. So you just take particular pages or particular mRNAs, which are the instructions for building these particular proteins that you need in a given cell. Just like you would take just the specific page that you need to build a particular furniture in every room. Okay, so the messenger RNA carries the information, partial information from the DNA that encodes for proteins. And proteins are the stuff that actually does the work in the cell. However, this is just one type of RNA. And in fact, only a small percentage of our genome encodes for messenger RNA, less than 2%. Much larger areas of the genome, of the DNA, transcribe to generate RNA, which is different, which is not messenger RNA, but has different functions. And some of these RNAs have functions, some are probably just a noise, although this is still argued a lot. One type of RNA that is not messenger RNA is regulatory RNAs. And there are different types of regulatory RNAs. The most well understood regulatory RNAs are small RNAs, which are called small RNAs because they're very short fragments of RNA. And what they do is they regulate gene activity, and they regulate this transmission of information from DNA to RNA and protein, eventually controlling which proteins will be made in our body, in each cell. So this is just the last introduction slide about this. What small RNAs do is these small RNAs can bind to specific mRNA molecules just by base pairing with them, by complementary interactions, and mediate a process that is called RNAi, or RNA interference, or gene silencing. RNA should normally not be double-stranded, unlike DNA. So if a small RNA finds a particular mRNA, and a double-strand RNA will be formed, and this double-strand RNA looks weird to the cell. It looks like a virus or something like that. So it leads to the degradation of the entire thing, of the mRNA. So the mRNA, following the binding of the small RNAs, stop being made, interrupting the production of the protein. And this is one way by which our cells regulate which genes will be expressed when and where, which is, of course, the secret of life. This process was discovered in worms and also in plants, and awarded its discoverers, Andrew Fire and Craig Mello, with the Nobel Prize in 2006, after it was discovered to be conserved in many different organisms, including humans. Okay. Now, in the original paper in nature where they found that double-strand RNA actually leads to the production of small RNAs and RNAi and gene silencing. In that same paper, they also found an additional thing which was missed by many, but it's equally important. It's very, very important. What they found is that silencing or RNAi works non-cell autonomously. So the RNA moves between different cells and can act in a distance. So first of all, what they've shown is that you can just inject double-strand RNA to a given somatic tissue in C. elegans, and that the silencing would not stay in this injected tissue, but it will also spread to other tissue. And later on, they showed that you can just feed worms with bacteria, this is their food, bacteria that produce double-strand RNA, and that the double-strand RNA that the bacteria make will lead to silencing of genes, of complementary genes, not only in the site of ingestion in the gut, but also in the rest of the body, because the silencing would spread to other tissues. And this also includes the germ cells. So the silencing would move from the soma, from the gut to the germline, to effectively breaching Weizmann's barrier, breaching this segregation of the soma from the germline. And over the years, we learned a lot about the mechanisms of how it happens. We still don't know everything, but we do know there are at least 8 different transporters that shuttle RNA between different cells in the worm, and that there are genes that are needed specifically for this non-cell autonomous regulatory effect, for this silencing in the distance, for transferring of RNA between cells. However, everything that I told you here in this slide about about small RNAs working in the distance is true for artificial small RNAs that are made synthetically. So if we make double-strand RNA synthetically or artificially, then it can induce this silencing effect that work in other tissues. We don't know whether natural endogenous small RNAs can do it as well, whether they also move between different tissues to regulate genes in other tissues. We have clues that endogenous natural small RNAs can do it, but it's more mysterious, and we don't know exactly which small RNAs do it. And this is something that we study in the lab. Okay, so when I started working in this field, what I wanted to know is, what's the physiological relevance of this? Is this actually happening, and does it also in natural circumstances, and does these small RNAs allow communication of the soma to the germline and transmission of memories between generations? And if so, which type of memories? What kind of memories or parental responses can be communicated to the next generation via these small RNAs? And at the time, I was just starting my postdoc. There wasn't anything known about this at all in the literature. And I was about to give up when I did what everyone does, I imagine, even if we don't say that we do it. And I just looked up Wikipedia after I gave up on the professional literature. And in the Wikipedia, in the part about the ecology of C. elegans, of the worm, there was a sentence that was since then removed that said that C. elegans is one of the few forms of life that doesn't have any natural virus that infects it. And I thought that perhaps this is because when viruses replicate, if it's an RNA virus, that it forms these double-strand RNA intermediates, which activate the RNAi system and lead to the destruction of the virus. Worms, the C. elegans don't have dedicated immune cells like we have T cells and B cells and NK cells. They don't have these. So they rely on RNAi for antiviral protection. And the hypothesis that I had was a very naive hypothesis. I thought that perhaps just like artificial small RNAs, these antiviral small RNAs would be passed between generations to protect the ones to confer this inherited small RNA immunity. Now, there wasn't any natural virus that infects C. elegans at the time. So I used an artificial virus that was tagged with green fluorescent protein, which is a protein, which is a gene that was isolated from jellyfish, and that glows in green and allows you to track certain molecules. And this was shown. It was shown that you can take this jellyfish gene, this GFP, green fluorescent protein gene, and put it in C. elegans, and the C. elegans would glow. This awarded Martin Schalfi with the Nobel Prize a few years earlier. So I used a fluorescent tagged virus that was used in the past to show that worms use RNAi to resist viruses. And so you see here on the left, you have, it's hard to see, but you have worms that have a functional RNAi system. They know how to create these small RNAs against viruses. And you see that they remain black. The virus cannot replicate, it cannot propagate in these worms because they use the RNAi system and small RNAs to destroy it. On the other hand, on the right, you see RNA mutants, worms that have a defective RNAi system. And these worms glow in green because the virus propagates in the absence of the RNAi system that destroys it. And from the wild type worms, so from the normal worms that have a functional RNAi system, you can also sequence antiviral small RNAs or virNAs. What I did was I used this system to see whether RNAi and small RNAs can confirm transgenerational memory of viruses and protect the worms. So I took these worms and I challenged them with a virus, the fluorescent virus. And since they have a functional RNAi system and they create small RNAs against the virus, the worms stay black and they destroy the virus. And then in the next generation, I disabled the RNAi system of the worms using very simple genetics, just crossing the worms with mutants that don't have RNAi. So that the next generation would be RNAi mutants. They can't protect themselves from the virus on their own. And I again challenged them with a virus. And then I wanted to see whether they can defend themselves or not. Since they don't have their own RNAi system that's functioning, they cannot mount a new RNAi response against the virus. So they should be green. The virus should replicate. That is unless they inherit small RNAs from their parents that will protect them. So it's a very, very simple experiment. And what we found in this experiment is that 100% of the worms are protected. So they inherit these small RNAs from their ancestors that destroy the virus. And not only that, this also transmits transgenerationally to additional generations. So this is a proof of transgenerational immunity mediated by small RNAs. And it's also the first time that heritable small RNAs were sequenced. Before that, it wasn't clear what's actually being inherited. Is it the small RNAs themselves, or perhaps the double-strand RNA, or perhaps the chromatin changes, because small RNAs can also enter the nucleus and lead to changes in the proteins that wrap the DNA and condense it. So it wasn't clear what's actually memorized the response transgenerationally. And in this paper, we showed for the first time that heritable small RNAs can be found, can be directly sequenced. Moreover, we found that the inheritance, the transgenerational inheritance, depends on a gene which encodes for a protein that's called RRF1, which is an RNA-dependent RNA polymerase, a gene that synthesizes more RNA, more small RNA molecules using RNA template, and amplifies the response transgenerationally so it won't be diluted. And I will speak more about this later on in the talk, but it's really important. Okay. While we use a completely artificial virus, and we didn't really do an infection, we challenged the worms using a different technique. Later on, Craig Mellow's lab, the person who won the Nobel Prize for discovering RNA, showed that another virus could also lead, when you infect the worms with a real virus, they also mount a transgenerational response against this virus, which is transmitted transgenerationally using small RNA. So the same mechanism, only in a more natural settings. And indeed, other labs have studied this as well. We wanted to know whether other responses can also transmit transgenerationally aside from viruses, from viral memories. And the first thing that we checked is the memory of starvation. And the reason that we wanted to know whether starvation leaves an imprint or a transgenerational trace on the progeny is that the effect of different diets and starvation in particular were studied also in mammals, including in humans. And these are harder studies to do, but the most famous study is the Dutch winter or the hunger winter, in which the Nazis in the Second World War starved for half a year a large part of the Netherlands, and people did a big epidemiological studies to characterize the effects that are seen in the descendants, in the children and grandchildren, and they saw that the children and grandchildren develop all kinds of traits that cannot be explained by changes in DNA, such as more diabetes and also schizophrenia. This was also studied by many different labs in rodents. Most of the studies that were done on mammals were following the next generations for only 2 generations. So we cannot exclude, it's very hard to exclude direct exposure of the offsprings to the environment, because the babies were already in utero while the mothers have been starved, so they were directly affected. And since the babies have their germline already, you can say that 2 generations are directly affected by the starvation. So we went and we wanted to study 3 generations to see whether this affects also progeny that were never affected by the original trigger, by the starvation. Moreover, in worms, it's very easy in comparison to distinguish nature from nurture, to know that this is not some environmental effect, but this is actually something biological that passes between generations. And worms, another advantage is that worms are genetically identical to one another. So you can rule out DNA changes very efficiently. What we did is we took worms and we starved them, or we kept them well fed. And then we sequenced their mRNAs and small RNAs, describing the changes that happen when they are starved. And then we let these worms produce babies and follow their children and grandchildren. Importantly, and we sequence also small RNAs and mRNAs from them. Importantly, when we starved the worms, we didn't starve them for too long. So we didn't starve them long enough so that some of them will die. The worms have a planned developmental arrest program that allows them to survive starvation. So they all survive. So we don't introduce selection into the equation. And what we saw in this experiment is that starving the parents leaves a mark on the progeny, even 3 generations later on, and that the transmission of the information depends on genes that produce small RNAs and transfer them transgenerationally, including a gene that's called HRD1, stands for heritable RNA deficient 1, which encodes for a protein that physically carries the small RNAs in the germline. And the small RNA pool and the gene expression pool of the progeny reflects the changes that occurred in the parents, and there are also physiological changes. The descendants live significantly longer, and later other groups have shown that they also survive better additional rounds of starvation and have other changes. For example, they develop slower. Since then, other labs have studied the effects of starvation and viral exposure on the next generations, and also other effects. For example, the effects of temperature, of high temperature, that also leaves a transgenerational, this also transgenerationally changes the descendants, and also transgenerational effects that occur in response to cultivation with pathogenic bacteria. This is very popular recently. A number of groups have shown that this affects the descendants. But I think that the most interesting thing to study now, this is the cutting edge, is to study whether the nervous system can also create transgenerational changes. Normally, when we think about memories, we think about memories that are held by the brain. And I very recently, just last week, I published a review about this, considering the possibility that brain activity can transmit transgenerationally. The reason that I think that effects that start in the nervous system are more interesting is that the nervous system, unlike any other tissue, is very good at planning and thinking ahead. So it really boggles your brain to think about, can the brain plan the fate of the next generation? I think this is very controversial and interesting. And to study this, before I will describe what we did to study this, I will say that there were studies from the 70s and 80s that are considered to be wrong today and also extremely controversial. I covered this in this review that I just published about transfer of memories between individuals and the possibility that memories lie in molecules. Normally, when we think about memory, we think about them as held in the synapses that connect different brain cells. However, in the 70s and 80s, it was popular to speculate that perhaps some memories are held in molecules. And people thought this is very interesting because then, in theory, you can manipulate these memories in a tube and even transfer them between individuals. And this started with the extremely controversial studies of James McConnell, who studied planaria. And planaria are the champions of regeneration. You can cut off their heads or their tails, and the part without the tail will regrow a new head after a few days. And they are also worms, but very different worms. They are flat worms. very different from C. elegans, which are roundworms. And they have a more developed brain. So they actually have, the brains actually have lobes. And they can encode more sophisticated neurons. And what McConnell said is that you can take these planaria, teach them something, and then chop off their heads, and that the new heads that will regrow will still contain the memories. Not only that, he also said that you can teach them something and then grind them up and feed them to naive worms, and the memories will transfer. And later, the most amazing thing is that they said that the fraction that actually transmit the memory from these chopped up worms is the fraction of the RNA. This is very interesting because these experiments were done long before we knew about regulatory small RNAs. However, people didn't believe him, and worse, eventually his studies stopped when he received a letter from the terrorist, the Unabomber, and exploded. So this interrupted his line of research. Other people studied the same things in rodents, but this was very controversial, and it led to a dead end. There wasn't any continuing of these studies. Nevertheless, it reached the general public and the popular media. There are comics books about it. There's even a Star Trek book about episode about it from 1984, where they described how memory is encoded in chains of RNA molecules. So this was very popular, although completely abandoned later on for 30 years or more. We want to know not whether McConnell is right or not, but whether small RNAs in C. elegans can actually transmit memories between generations from the brain. So what we did is I won't get into the details, but we tracked small RNAs that are made specifically in the nervous system of the worms. And we showed that changing the levels or the production of small RNAs in the nervous system, in the brain of the worms, lead to transgenerational changes in the small RNAs in the germline and transgenerational regulation of genes even 3 generations down the road. So the ancestor's brain, using small RNAs, control the genes of the progeny for multiple generations. This depends again on this heritable RNA deficient 1 protein, which carries small RNAs physically in the germline. And we have some clues into how the brains connect with the germline, although much of it is still mysterious. We don't know exactly which type of molecules move and how. Nevertheless, we found that this transgenerational transmission from the brain to the germline is crucial for controlling the progeny's behavior, their capacity to find food. We found that RNA mutants, certain RNA mutants that are called RD4, can't find food. They can't do chemotaxis, can't follow a chemical gradient when it's slightly hot, in slightly stressful temperatures, unlike wild-type worms, which do it very efficiently. However, if these RNA mutants derive from wild-type parents, or from parents or grandparents, or even grand-grandparents that had functional small RNA production in their nervous system, then the RNA mutants can find food just fine. Not as good as normal worms, but much better, which means that the nervous system of the ancestors can control the capacity of the descendants, even 3 generations down the road, to find food, to behave. And we pinpoint one gene. We found one gene that is controlled transgenerationally in the germline by the ancestor's brain, which is called SAGE2, which is responsible for this heritable control over behavior. So this is the beginning of showing that the brain can actually communicate to control behavior with the next generations. The last topic that I will discuss really very, very briefly in 2 or 3 minutes is whether there are any rules to this transgenerational inheritance. Normally when people think, or also in the past when people thought about epigenetic effects and transmit transgenerationally, many people speculated they just passively dilute or go away. So for example, if you experience some trauma or some change, then the descendants will have a less severe effects and gradually it will go away. Also, when people describe this heritable RNA effect, this is how normally they thought of it. However, we knew that this is not possible because every worm produces something like 250 babies. So after 4 generations, and these effects last 4 generations, typically transgenerational effect in C. elegans lasts 3 to 5 generations. So after 4 generations, the dilution factor would just be enormous because since each worm produces 250 babies, it will be 250 times 250 times 250. After 4 generations, it's 4 billion, which is completely homeopathic and can never work. Okay. Moreover, we found the transgenerational inheritance in C. elegans depends on amplification of the small RNAs by these RNA-dependent RNA polymerases that synthesize new small RNAs in every generation using mRNA template. So if they can make new small RNAs in every generation, why does it ever stop? Why does it stop after 3 to 5 generations? We think that it stops because it's programmed to stop, because it's not good to remember everything that your parents did. Something you need to adapt and change. And in fact, we identified a mechanism, a sort of a negative feedback loop that turns off the inheritance after a few generations that we called the transgenerational timer. And this is a clock made up of genes, of proteins encoded by genes that we dubbed modified transgenerational epigenetic kinetic genes, or MOTEC in short. Those of you who speak Hebrew know that MOTEC means sweetheart in Hebrew. And if you mutate these MOTEC genes, you get responses that have a very different duration. So instead of lasting for just 3 to 5 generations, you have mutants, MOTEC mutants, where the response lasts tens of generations or even hundreds of generations, so that they appear to be completely stable. And we found that stress, that subjecting the progeny to stress reset ancestral responses that are contrasting. So this is the heritable responses are plastic. We very recently published it just last month. And also, and again, I won't get into it because it's more complicated, we identified 3 simple rules that explain how transgenerational responses segregate in the populations. You know that genetic effects segregate based on the rules that Mendel identified more than 150 years ago, which basically just reflect the process of meiosis. Transgenerational small RNA responses segregates not based on Mendelian genetics, but based on other rules, 3 simple rules that allow you to understand how the response would segregate between genetically identical individuals transgenerationally. And this allows you to predict for how long the response would last and which individual will have a strong response and which individual will have a weak response. And there's also one protein which is a major regulator of stress called HSF1, which creates diversity, intentional diversity between the descendants, just like there are mechanisms for creating genetic diversity as a sort of a bat hedging strategy. So with this, I will stop this is my last slide. I would just say that what we think now is that at least in worms, and we don't know what happens in other organisms, RNA was always in the background, inherited, and we just didn't see it because it's harder to study, because it changes also in response to the environment. And when people sequenced the human genome 20 years ago, we were surprised to see that humans and worms have pretty much the same number of genes, while corn has much more genes, many more genes. So of course, it's how you use your genes that matters, not how many genes you have. And transgenerational effects, mediated in part by small RNAs, can perhaps explain the differences between different organisms and the heritability of certain traits. And with this, I would end and just thank everyone in the lab and you for listening. Thank you very much.
Many thanks, Oded, for giving a very clear and amazing talk of all those kind of discoveries from initially going to Wikipedia. And who'd have thought that a trip to Wikipedia, I guess it's serendipitous discovery, and the stepwise, very simple transition that you took to actually uncover all those different stages. And it seems that it's going to be a never-ending story, I think, of these discoveries, one after the other. And incidentally, in really top quality, the best top quality journals. So that's amazing. So I'm going to the next phase of this is to get responses from to your presentation from Tony Hannon and Elsha Lawson-Boyd. We'll each put a different lens on the topic you discussed in your talk and potentially ask questions. This will run for about 10, 15, 20 minutes, depending. So first, I'm going to introduce Professor Tony Hannon, who received his undergrad training and PhD from the University of Sydney. He was then awarded a Nuffield Medical Fellowship at the University of Oxford, where he subsequently held other research positions before returning to Australia to establish a laboratory at the Florey Institute. He has received various fellowships and awards and is currently an HMRC Principal Research Fellow and Theme Leader at the Florey Institute, University of Melbourne. His laboratory at the Florey explores how genes and the environment combine their experience-dependent plasticity in the healthy and diseased brain. I should also add that he's very active and campaigns for funding and respect for scientific research, for medical researchers and other researchers throughout Australia. So it's a great advocate for medical research. So Tony, over to you.
Great. Thank you, Jeff, and the organisers, and thank you, Oded, for that fantastic talk. It's so exciting and just such wonderful science. So, yeah, I'm coming from the other perspective where we're working with mice, and I know there's this rivalry between the worm guys and the rodent guys. And so we're thinking about your worm work from the perspective of mice and men, of mice and humans, and how in these different species, from invertebrates like C.elegans to mice and humans, how we can fit this into a framework overlaid on Darwinian evolution. So the idea of how you transmit information between generations, a very slow version of gene mutation and natural selection, but the idea of this almost Lamarckian evolution, the way it's overlaid. And just as an aside, those who aren't aware, Jean-Baptiste Lamarck was, when he put forward hundreds of years ago, his theorem, who's largely ridiculed, whereas Charles Darwin was a bit of a fan of Lamarck. And one of the lessons there is people who ridiculed Lamarck would say, well, you know, the giraffes reach up to the higher leaves, and so their offspring have longer necks, ha. Wasn't, you know, Lamarck an idiot? And so one of the lessons here is that you should never dismiss a new theory or hypothesis out of hand, because I think what's coming through now is this kind of Lamarckian evolution overlaid on Darwinian evolution, and the idea that they're compatible, because clearly what you've discovered in worms and what we and others are working on in mice and in humans, other vertebrates, is clearly evolved through gene mutations and natural selection to give you these transgenerational epigenetic inheritance mechanisms, which may act via non-coding RNAs, but have evolved via, we assume, random DNA mutations, but there's clearly other evolutionary mechanisms at play. And that in itself is interesting. So I guess for this audience, you know, what we think about is the relevance of this to humans. And there's increasing evidence from our perspective, the work we do on mouse fathers and sperm. There's been some lovely studies done, for example, these obese men who were resistant to losing weight, so they had surgery to lose weight, and they found that the sperm epigenetics, including non-coding RNAs, that were abnormal in the obese men as post-surgery, as they started to move back towards normal. These, you know, epigenetic signals in the sperm started to move back towards normal. That's one of the studies I think is compelling, that suggests that this is happening in humans. So I guess for us, thinking about mice and humans and other vertebrates, the big questions from our perspective is how this information can be encompassed in the sperm on the one hand, and then post fertilization in mammals, how this information may actually control something as complex as brain development and brain function. And so there is a controversial paper, which I'm actually quite skeptical about, many years ago, about 2014 in mice, where they claimed that this olfactory memory was transferred from male mice to offspring. And part of the problem, apart from the actual data in that paper, which is problematic, but part of the other problem is the philosophical problem. How would brain memories, if we think about that, how would that information be transferred from the brain, that level of complexity, through the bloodstream, through the Weizmann barrier, which we accept in mice, it can be breached, but it's an informational problem. How you take something as complex as brain experience, how you might move that down to germ cells and encode that information and then transfer that information to the next generation. So there's some of the big questions we're thinking about that I would lead off the discussion. I don't know whether Oded wants to respond to that.
Yeah, I would. These are all on point. Great points. I would. You said a lot of things, so I will try to remember. And please remind me. But first of all, with regard to Lamarck, you're totally right that Darwin was a Lamarckist, and he even wrote about it in The Origin of the Species. He had this pangenesis theory about the body, the somatic cells communicating with the germ cells. But moreover, both of them, Lamarck and Darwin, just didn't know genetics, so they were both very wrong. The problem with Lamarck is not the inheritance of acquired traits; it's more the teleological nature of his philosophy. So when I'm talking about transgenerational effects, parentally carry over the effect, I think it's not necessarily Lamarckian, because Lamarckian is a broader word. With regard to the last point that you said, so first of all, I don't see it as a rivalry with the mice people. I like mice, I like humans too, and I think that for us to justify our existence, it does need to be relevant also to higher organisms. That's very important for me as well. And I think that, there are some differences obviously between worms and mice or humans, primarily the way that the worms define their germline. So the germline in C.elegans is continuous, while it is being defined later on in development in mice, which is a problem. But it's not a problem you can't, deal with theoretically as evolution. It could be worked. And another thing is that the worms, unlike mice, have these RNA-dependent RNA polymerase, which amplify the small RNAs transgenerational. So while there are many examples of effects that seem convincing in mammals for inheritance, most of them were studied for one generation and not transgeneration, not for longer generation. That does not mean that it's not possible to transmit information transgenerationally in humans as well for multiple generations. It would just require additional mechanism. For example, any other feed-forward mechanisms that will maintain the memory, like DNA methylation, which is also connected to small RNAs, or changing in chromatin, or any other sense-antisense mechanism that we can maintain it, like ping-pong cycles that were shown in other organisms. So I think that it can all be compatible. With regard to the last point that you said, that's very, very important about how could a memory, when you normally think about memory, something that resides in a specific neuronal architecture, involves many neurons and so on, how can it transmit through the bottleneck of a single fertilized egg to affect the development of the nervous system in the next generation? In worms, the evidence that we have from our studies is that in fact there's a reverse Weizmann barrier. We don't know whether it is completely, unpermeable, but what we see is that if you regulate genes transgenerationally, if you treat the worms with RNAi to target genes, the genes that get regulated transgenerationally are germline expressed genes. The somatic genes get re-expressed, and this is true except for a few exceptions. And nevertheless, the nervous system can produce transgenerational effects that affect behavior in the next generation. So how? We think that you can regulate the behavior of the next generation by affecting germ cells, physiology, because the germline communicates with the rest of the body in many different ways. It secretes hormones and other molecules and also affect development later on. So for example, in our case, where we show that the nervous system of the worms affect the capacity to search food in the next generation, this is happening owing to the regulation of a germline gene that's called SAGE2, which somehow affects the behavior later on. We also have a new preprint that we just published a few months ago, where we show that when you subject the worms to stress, small RNAs that regulate the sperm prevent the sperm from fusing with the oocyte. And when the sperm doesn't fuse with the oocyte, the worm thinks that it doesn't have any sperm, so it secretes a pheromone that attracts males, leading to mating. So again, you affect behavior in the end, which is mating, but this happens totally indirectly by regulating germline physiology. So I think that while some mechanisms that communicate complex gene regulatory effects or neuronal architectures can also be imagined, because at the end, the structure of the nervous system is encoded in the genes to some degree. So you can regulate development and also the development of the brain. Some effects that would affect behavior would be indirect and nevertheless extremely interesting. So this is what I wanted to say.
And can I ask, do you think these mechanisms in worms evolved independently of the mechanisms in mice and humans to the extent that they are transgenerational?
Right. The simple answer, I have no idea. But I think that I do think, and this is again total speculation, that it's too good to give up. So I imagine that some of it exists in this form or another, whether conserved or maybe it's a convergent evolution. But I believe it makes sense to transmit this information also in other organisms. However, I would say that this depends also on the particular response. One reason why transgeneration inheritance in worms makes so much sense is that the generation time is so short. So it's just 3 days. So the likelihood that the parent's environment would resemble the offspring's environment is very high. So you're preparing them for something relevant. While with humans, maybe my kids will go to live in Australia and it will be a post-COVID world and everything will be different. So maybe it will be different. On the other hand, some stimuli or some challenges that we meet, like viruses, we meet them over and over. So perhaps it would make sense, even in organisms with a long generation time, to transmit information transgenerationally. It depends on the story.
And it all evolved before we had aeroplanes. Our ancestors didn't move very far.
That's also true. Yeah.
Okay, thanks, Tony, for your response, and Oded for addressing those questions. Next, I'd like to introduce Elsha Lawson-Boyd, who's a PhD candidate from Deakin University, Melbourne. Using feminist theory in tandem with qualitative interviews, Elsha's doctoral research engages with the emerging field of neuroepigenetics, paying attention to narratives of inherited trauma, along with Conrad Waddington's idea of epigenetic landscapes. This study explores neurological plasticity through the lens of absorption, which conceptually holds together the moment of flexibility and stability. As a strong believer in collaborative work across the biological and social sciences, Elsha uses interviews as a means to engage with neuroepigenetic researchers in countries like Australia, Canada, Switzerland, Singapore, and North America. So over to you, Elsha.
Great, thanks very much for the introduction, Jeff. And might I say that was a hard act to follow, so I am going to do my best to come into this conversation with quite a different perspective. As Jeff explained, my work is based in feminist theory, science and technology studies. So my background is mainly in the social sciences, although I have a more, I suppose, casual engagement with biology. I was really interested in your talk, and thank you so much for explaining so well what seems to me and what I'm sure seems to me, my colleagues, is incredibly complicated scope, scaling inwards into the body. And our role, I suppose, is to do the kind of scaling outwards and see what can be transferred or not between different scales. So my first thought really was there seems to be a lot of hype and hope around epigenetics from an outsider's perspective. Social scientists write about this opportunity for age-old binaries such as the nature-nurture, culture biology, but also sex, gender, male-female, and even what you were talking about with brain and body, or mind and body. So on the outside, it seems like this is very set knowledge. It's already, the horse is already bolted. It's already had such positive reception from many different lay communities. And I found that especially interesting with notions of intergenerational trauma, and when I've spoken with a number of different scientists, they've said they've had their own positive feedback from people who have just read about this and it's such a validating source of knowledge for them. But I'd really like to hear more about what it's like on the inside, because you're talking about, dogmatic ideas of genetics, and how has it been for you in this field, coming in with, what you're talking about is quite controversial ideas. So there's that. And there was also something that came to mind when you were talking about imprinting, where, it runs on this neuroplastic idea that, unlike genetics, epigenetics can be changed, it can be reversed, it can even be removed. And that was a key part of my interviews. But a kind of conundrum that seemed to come up was what's actually being removed. If we're talking about, intergenerational trauma, or we're talking about stress, how do we conceptually separate what is consequential, negatively consequential, and what is memory? And how do we reconcile that conundrum? And one other thought, just before I don't want to overload you with information, but there was a fantastic paper by my colleagues, Bruce Mueller and Martha Kenny. They actually called it Rats and Women. And it's about the kind of gendered assumptions that have made their way into epigenetic epistemologies, research designs, as well as the sort of questions that researchers are asking, which was a fabulous contribution. And it provides a not just a feminist critique, but a feminist engagement with the science itself. So I was wondering if perhaps you had any thoughts on future collaborations, or what does sort of interdisciplinary work look like for you? Yeah, thank you.
Thanks. These are great comments. I have to say, first of all, I didn't know about this paper about rats and women. I should definitely read it. It sounds very interesting. I never thought about it from a feminist perspective, or as something that affects gender theory. But it is interesting to think about it. I should be familiarized with the literature, because I'm unaware. I would say, however, that we know that historically, epigenetics and epigenetic inheritance is associated with the left, with the perspective of people who believe in change, and that it's not all predestined. And this sounds like a good thing, and it could be a good thing. However, any theory, whether it's Darwinian evolution or Lamarckian evolution, can be abused. And as we know very well, the theoretical capacity to change was abused in the Soviet Union by Lysenko and the people who said that everything can be changed, and it's all plastic and reversible, and this led to massive starvation and lots of problems. So, but it's true, and also I wouldn't say that this is accepted globally by everyone. I think, as you probably in your interviews, I'm sure you encountered people who study mice that have a very hard time getting these ideas across. I think with C.elegans, we're lucky because people see it as a sort of a curiosity. These worms are different, so maybe it's possible, and we don't get too much backlash. But it's much harder for people who study mammals. And another thing that we have to deal with, and this happens in the beginning, it happens less now, I don't know why, but when I published my first paper in this topic, I got emails from creationists and all kinds of people saying that this goes against the theory of evolution, when in fact it's just like Tony said, it's totally compatible. It's something that you add on to the normal framework, and it just can perhaps explain more. But these are the type of things. So I don't know if I addressed everything that you said, but I think that it's very important to connect it with the social studies, and to also understand this. And like everything, there's tons of fake news, and epigenetics. The word epigenetic has been used for everything. For me, epigenetics are changes that continue, whether it's after cell division or transgenerationally, independently of DNA changes. That's my definition. But people use it in different ways. Some people talk about, epigenetic anti-wrinkle creams, and it's everywhere, this term. And so we have a lot of misinformation to deal with, and and not enough dialogue with people from other fields and from social studies, so that the public will be properly aware.
Mm-hmm. Yeah, and that's such an interesting point about you can't really separate the politics from the science, right? Like it's that idea of plasticity and malleability as being progressive in some sense, but can also sort of have these unanticipated consequences politically. Yeah, no, thank you. I was also going to mention, I wanted to ask you about your research, because with C.elegans, I'm imagining this is basic research. It's not translational research. Yeah, because I was wondering, I mean, one of my contributions in my own PhD is talking about cross-disciplinary engagement to think more about agents. And, you know, when I talk with scientists, the agents, you know, neural transmitters or epigenetic markers, but in a social science perspective, our agents are, you know, geopolitical frameworks or all the like outside the body. So yeah, I was wondering, like, how do you see your kind of research? I know you've touched on it a little bit, but how do you see that moving into human research and human studies?
Yeah, so luckily for me, I don't have to justify that too much so far, but I think there could be implications later on to treatment that's further down the road. But for example, if the concept that really the environment changes the pool of heritable small RNAs, that this can predict all kinds of traits, then it could find its way to diagnostics first. Just like now, every couple that goes to have a baby does genetic screenings, you could also characterize the pool of heritable small RNAs. And the beauty is that this pool would also be changing in response to your body, to your activities. So perhaps the father will be told to run on a treadmill first or something like this. So that's the potential. We're not there yet, but it could reach this some other way. But I'm personally not focused on that.
Mm-hmm. No, thank you.
I was on mute. Thanks, Elsha, and thanks, Oded. We have some really interesting questions, which I will go through now from the audience. So, first question from Emma Koval from Deakin. She says, "Wow, RNAs are great at fighting back viruses. Do they also work on bacteria? Otherwise, why did other organisms bother to evolve an immune system?"
Yeah, so great question. First of all, I would say that what we've shown with transgenerational antiviral immunity is conceptually very similar to the concept of CRISPR, this antiviral immunity in bacteria that works via different molecules, also small RNAs, and also involves a step in the DNA of incorporation into the DNA. In worms, recently there have been a couple of papers about transgenerational immunity to bacteria being, not from my lab, being mediated by heritable small RNAs. There, the small RNAs don't act directly on the bacteria to destroy them. What they do is they somehow control an avoidance behavior that gets the worms to move away from this pathogenic bacteria and survive better. So this was shown also by a number of different groups, and they argue a little bit about the mechanism and for how long does it last, but there are a few good examples now that show that this also allows transgenerational immunity against bacteria in worms.
Thanks, Oded. Question from Mate Varga. What would be the adaptive value of transmitting an epigenetic memory in a species with around 25 years of generation time, e.g. humans? I think you touched on that a bit earlier in one of your answers.
Yeah, right. I would just say briefly, very briefly, it's not good for everything, but if we're talking about viruses that we encounter over and over, then perhaps, and of course I'm just speculating, I don't say that this is happening, this is just theory, perhaps this would be beneficial. We don't know why these pandemics erupt every 100 years. You know, perhaps that's a few generations. I'm just guessing, you know. But we are meeting the same viruses over and over. And again, this is a disclaimer. I didn't say this. This was just theory.
I might add to that, to answer that question. When you think about the factors and in rodents, and some of it's been, evidence from humans as well, like diet. Think about our ancestors as we started to move into different environments, our ancestors moving around Africa and up towards Europe. So over that time period, they would have been shifting environments by migrating. And so as you migrate into different environments with different food availabilities, fats and sugars and proteins and other foods, then that potentially would have. The other thing is, in terms of our the rate of evolution, if some of this did start in the common ancestors of, say, rodents and humans, in a mammalian ancestor, which much shorter timelines, then whether some of this is a carryover from a common mammalian or vertebrate ancestor, which would have had shorter, generation gaps. And so there might be elements of this which are carried over, but still have some advantage. So we don't know whether it's a carryover from our common ancestors. And we also obviously don't know the extent in humans. We just haven't done enough of the human studies to really have a strong feeling. But the rodent data is pretty strong. But then again, they, can breed every couple of months. So it's a very different timeline.
I would just say that also in worms, we don't know really about the ecological relevance of our studies because we don't know enough about the ecology of the worm. So what we do, is very artificial still.
There's a new meme in there. Somewhere, what's the ecological relevance? Remark at the start of that meme. So next question from Monica Wesley. Any evidence that shows small RNA controls transgenerational immune cell functional control? Also, can small RNA input to the immune system come from commensal bacteria in the gut? Thoughts on type 1 diabetes?
It's a good question, but I don't know. Maybe Tony would know. I don't know the answer. I don't know whether any interesting studies in this, perhaps, but I'm unaware.
Yeah, I can speak with our one study last year where with collaborators in Melbourne, we studied paternal toxoplasma infection. So it was a Cell Reports paper. And so the fathers who'd been infected with toxoplasma, and then we waited, the usual about 4 weeks, and then bred with control females. We showed both changes in the small non-coding RNAs in the sperm of the fathers who'd been infected with toxoplasma, and then changes in the offspring, including mainly behavior is what we focused on, and even then micro-injected the small non-coding RNAs into fertilized mouse eggs and showed recapitulation of at least part of that phenotype. So we have got evidence of paternal infection affecting these non-coding RNAs in the sperm and passing information to the next generation.
I apologize. I knew this study. We also worked on toxoplasma with very different reasons. So that's wonderful. Thank you.
Thanks, both of you, for that. There's a question from Sumit Garai. Is it true in a way to think that simpler animals, such as worms, have neuronal circuits, development of which is highly dependent on genetic and epigenetic landscape of the organism, but more complex animals, such as humans, have way more neurons, I like that way with 2 A's, way more neurons than genes. So the brains might develop in a more gene-independent way, e.g. neuronal group selection. So Sumit's question is, maybe simpler organisms, do you think simpler organisms are more vulnerable to their brains changing by things like RNAi than complex ones?
It's a good point, and it might be true. I mean, there's this concept of neoteny. Humans and more sophisticated organisms take longer to develop and so that their brain will be more plastic and can learn more, sculpt themselves based on the environment. And this is true. Perhaps this could also affect the capacity to be regulated transgenerationally, but we don't know whether this is the case or not.
And I'd probably chip in there in terms of, this idea that with evolution and big-brained organisms like humans, that compared to some others, we don't have many more genes, but we have a lot more of these non-coding RNAs and a lot more of that informational RNA in terms of the developmental algorithm. But what you have to remember, and this is Jeff's area here, is that monozygotic twins that have the same genome, by birth, they look identical, and their brains, if you image them, are very, very similar, with a very similar number of cells. So, there's kind of evidence that even in humans, if you start with the same genome, by birth, this developmental algorithm can generate pretty similar brains, and then environment starts to shift them so that, on some traits, monozygotic twins might be 50% discordant, for example. But, things like, twin studies are very powerful in terms of saying that genetics can still guide the evolution and the development of a very complex brain like humans. But we're interested in this concept of decanalization, that the brain's experience expectant. And so when a brain develops in an environment that it doesn't expect or gets a major unexpected instruction, you can decanalize development, including brain development. And we've put forward a theory that might contribute to autism spectrum disorder and schizophrenia and developmental disorders of the brain.
I think my take on that, and what I've seen of twins, is you look at the use twin models to break down the components of variation. What makes us what we are? Environment's in there. It's a small piece of the pie. The next biggest piece of the pie is genetics, and the biggest piece of the pie is basically random chance. Or look at another way, development. So when two twins are born, my guess is that their brains, if you look close enough, would be subtly different. And there's a number of, even from anecdotal, from parents that have told me that they're identical twins, as soon as they're born, they have different temperaments. And so you have that stochasticity or plasticity, natural variation built in that's outside genetics and potentially outside epigenetics. And so I think it's something that all multicellular organisms, maybe even some single cell organisms have, is that, it's very, I don't know, maybe Oded, you can comment on this. We talk about, that animals differ, that complex organisms differ because it's developmental stochasticity, but does that same thing happen within C. elegans too?
A little bit. So they would develop, some things would be very fixed, like the number of cells.
Yeah.
Number of cells would be very fixed, but if you do a behavioral assay on C. elegans, a chemotaxis assay, you will get variability in the population, and 80% of the worms will go there, while 20% won't. We studied a little bit the reasons for the diversity in epigenetic inheritance in a recent paper, but we obviously don't understand it fully.
Okay, thanks.
And I guess the other thing to add to your point, Jeff, when you talk about stochastic mechanisms and random changes, when you get outside of the brain and think about the body and craniofacial development and the face, these monozygotic twins emerge more or less, you can't distinguish them, they're so similar, and yet craniofacial development and face development is very complex, it's not simple. And so, this genetic algorithm is precise enough to produce two bodies that are pretty much indistinguishable at birth. And in fact, often these monozygotic twins are indistinguishable physically for a long time. So yes, in the brain, there might be more noise and more stochastic processes, but there still seems to be a lot of precision when you look how similar identical twins really are.
Okay. Thanks, Tony. Question from Mireille Uchao. On the olfactory study in mice, is there division between memories held in the brain and the body? Do trauma studies show that memory resides partly in the body? Interesting question.
I think this is a question for Tony, perhaps, or
yeah, I'm happy to answer that. The one paper that claimed that these olfactory memories were being transmitted very specifically to sperm epigenetics in the next generation, to my knowledge, haven't been replicated, extremely controversial, published about 7 years ago. And that really was implying, and it was an informational problem, the idea that these very specific olfactory memories were then being transmitted from the brain down to these hundreds of olfactory genes and leaving methylation marks and so on You know, I think if you make extraordinary claims, you've got to provide extraordinary evidence. And so, you know, even though there are wonderful papers published, there's also sometimes papers published that don't get replicated, and we have to be open to that in terms of the need for replication and so on. So I think in terms of information in mice and humans going from the brain to the germ cells, absolutely it can happen, but it's just a case of what precision of information. If these molecules are being released, say, from the brain and getting down into the germ cells, what precision of information could they convey that related to the experience? And therefore, it's more straightforward when you think about stress and diet and exercise that we've worked on as well, and even infection, you know, in terms of what kind of information might travel through the bloodstream and then transform the sperm epigenome and then transform the embryo. But when you're talking about information carried by neurons in the brain, at some point, turning that into information in male germ cells, for example, you've got to have a mechanism that can explain that movement of information around the body, really.
Right, I totally agree. I also think always about these general responses that transfer transgeneration, like, you know, an inflammation, higher capacity to learn, or something like that. That's true. However, you could imagine, I'm not saying that they showed it in this paper, but you could imagine, let's say in C.elegans, where in C.elegans, I mean, the worms distinguish thousands of odors, and they do it using a very small number of sensory neurons. You know, when it comes to appetitive odors are sensed by 4 neurons. And which express many olfactory receptors. So in theory, you could regulate the levels of one particular receptor transgenerationally to confer a very specific change in the sensing of a particular odor. But this has never been shown. What has been shown with a high specificity or resolution, which is also, I think, because it's very new, it's just a preprint, it remains to be replicated and so on. But there's a paper from Colin Murphy where she showed that a bacterial RNA moves from the bacteria to the worm and regulates a particular gene in the nervous system of the worm to affect its avoidance of one particular bacteria and not a different type of bacteria. So that's but that's very new. And again, you know, it remains to be seen how this works. But this is such extraordinary claim that has been made recently, just a preprint, and we'll see.
Yeah, I agree. We need to be open-minded, and in worms, there's things that, like you said, the simplicity means the transfer of information is more feasible. When you're talking about a really complex memory in a mammalian brain, the complexity of that memory.
Right, although also in worms, RNA, small RNAs are very inefficient in reaching the brain. So I talked about this reverse Weizmann barrier. In fact, neuronal genes you can't silence them with RNA. It doesn't reach the brain, except, you know, in special mutants where RNA is much stronger and so on. So it's not clear exactly how it works. In her case, it could be that this bacterial RNA is very special, but normal small RNAs do not reach the brain in C.elegans. And C.elegans brains doesn't express SID1, you know, this transporter of small RNA. So we don't know if the exact same mechanism, what's going on there.
Excellent. One question from Karina Troitz. Following up on Mirai's question and Elsha's prompt about neural body distinction, I'm curious about how these distinctions are maintained or troubled, especially in different species studies. Who wants to answer this one?
I hope I really understand the question. I think that the question is, can you separate a neuronal response or something, a thought, a neuronal process from a bodily response? And I think that if this is the meaning, then I think that in the end you really can't, because I mean, a thought would affect the physiology of the brain and therefore will lead to biological changes at the end. However, I do think that it's a very important, outstanding question to see whether it's enough to just think about something to generate a response. So if you traumatize a mouse by forcing stress on it, then you're also changing other things. You could imagine just activating a neuron with light or something to really have a more chirurgical change, but this is more difficult. And I think this is the future. I think things like that should be done. And perhaps on the things that like that I'm unaware of in mice are being done to just, specifically make sure that it starts in the neurons. In our study, what we did is we just manipulated small RNA expression in neurons. However, this is also biological. It's not that the worms thought about something and this triggered the change.
Yeah, look, I get, there's an example, I think it was directly following up your worm study, Oded. A colleague up in Brisbane published this paper where they injected a human microRNA into the mouse brain.
Yeah, John Berry. I know this John Berry one.
Yeah. And so, and then they showed it actually transferred down into the testes of the mice. So they showed that transfer. Now, that's just a microRNA, but I think the key point, getting back to the question, and we're fascinated by this, is this complex bidirectional interplay between the brain and the body. And a great example is the gut microbiome. You've got trillions of bacteria in humans that are talking to the brain, and it appears that the brain, through the vagal nerve and other mechanisms, you've got a sympathetic and parasympathetic nervous system. So your brain can actually change. Like if you get stressed, your heart rate changes and you sweat and you get all these fright and flight responses. It's a clear demonstration of how brain activity can immediately change a whole range of organs. And then the other way up, all these changes in organs affecting the brain. And we're interested, there's a whole bunch of brain disorders that seem to involve the immune system, that might involve change in the gut and gut microbiota talking to the brain, and vice versa. So starting to think about, and the way in which environmental factors could affect the brain via the body, so we're very interested in that continuous crosstalk between the brain and the body. And we can't think of them really as different. They're very much intertwined.
Thanks, Tony. And that brings us all to the end of this talk. So I'd like to briefly thank Oded again for such a great talk and a great discussion, and many thanks to Elsha and Tony and all those who asked questions. We're really enriching this last hour and a half. I think we've all learned a lot. So many thanks, everyone.
Thank you very much.
Thanks very much, Jeff.
Right.
Thanks, Tony.